Hi, my name's Drew and I'm a PhD candidate at the Australian Laboratory for Emerging Contaminants at the University of Melbourne. Lord Howe Island is a small island off the coast of New South Wales between Australia and New Zealand. The island is an important breeding ground for the flesh-footed and wedge-tailed shearwaters.
So Lord Howe Island has two populations of shearwaters, the wedge-tailed shearwater, which is slightly smaller and, as the name suggests, has a wedge tail, and the flesh-footed shearwater, which has flesh-coloured feet. And there are approximately 20,000 flesh-footed and about 40,000 wedge-tailed.
Another significant impact on the populations of shearwaters on Lord Howe Island has been linked to the ingestion of marine debris.
I've been working on Lord Howe Island almost continuously since about 2007. And over that time, we've recorded a decline in the flesh-footed shearwater population, while wedge-tailed shearwaters appear to be relatively stable, if not increasing.
We have found up to 90% of fledgling shearwaters on the island have plastics in their intestinal tracts.
To put into words how exactly you feel when you encounter a bird that has very clearly died from the ingestion of plastic. There's just so much plastic and the tragedy that is unfolding literally in your hands.
As apex marine predators, shearwaters are exposed to trace metals such as mercury, copper, zinc, arsenic and cadmium, leading to increased concentrations in their bodies.
We've been studying trace element concentrations in the flesh-footed shearwater primarily over the last 10 years, and we've discovered that the concentrations are extremely high in this species, potentially exceeding a number of thresholds for things like lead and arsenic that would be cause for concern for the health of the species.
As these metals can sorb to plastic, we expect some of the bird's exposure to be from ingested marine debris. In addition to trace metals, persistent organic pollutants, or POPs, have been shown to also accumulate in biota and on the surface of plastic, both of which are thought to contribute to the overall exposure of POPs in the marine birds.
Persistent organic pollutants are chemicals that persist in the environment, and then they also bioaccumulate in wildlife and humans.
Today, we are concerned with other halogenated organic compounds. Per and polyfluoroalkyl substances, or PFAS, are a family of over 4,700 individual compounds.
You probably know some of the products that PFAS were incorporated, and this includes Gore-Tex, Teflon, and Scotchgard. These are the most common PFASs that were found in your home. So PFAS are a synthetic group of chemicals that have been used since the 50s for a range of industrial and domestic purposes. The most famous of these are PFAS and PFOA.
Once in the environment, they're extremely persistent, mobile, and potentially toxic. These criteria have led the compounds being internationally recognised as persistent organic pollutants.
The industry started phasing out these chemicals, and unfortunately what happened was that a family of chemicals, of fluorinated synthetic chemicals, has cropped up to replace them.
In this study, we report the concentrations of 45 PFASs in 2 species of juvenile marine seabirds from Lord Howe Island. Correlations between PFAS concentrations, body morphometrics, and plastic ingestion were compared to determine the potential ecotoxicological impacts to juvenile shearwaters. This is the first study of the occurrence of PFASs in seabirds from the Southern Hemisphere. Between April and May 2019, 33 flesh-footed shearwaters and 9 wedge-tailed shearwater fledglings were collected after they had died on the beach. The intestinal tracts of the birds were studied, and the number of plastic pieces and the total plastic mass was recorded for each bird. Back in the lab at Melbourne, the livers were prepared for analysis first by homogenization, the addition of mass-labelled internal standards, and the extraction of PFAS with acetonitrile. After cleanup, the extracts were analysed by LCMS, which allowed us to detect PFAS concentrations in the order of tens of picograms per gram of liver. Plastic was found in 79% of the birds, and one individual was found with 23 centimetres of nylon rope in its gizzard, weighing around 7 grams in total. This is consistent with previous studies conducted on the population between 2011 and 2018. Of the 45 PFASs monitored in this study, 16 were detected from 5 classes, including perfluorosulfonic acids, perfluorocarboxylic acids, fluorotelomere carboxylic acids, perfluorophosphinic acids, and perfluoroalkane sulfonyl fluorides. Compounds like 7,3-FTCA and 6,6-PFPIA were an interesting discovery, but these were only detected in a handful of birds. On the other hand, long-chain PFSAs and PFCAs were detected in over 50% of individuals. PFAS, the C8-PFSA, and PFNA, PFDA, PFUDA, and PFTRDA, which are the C9, C10, C11, and C13 PFCAs. As many previous studies have found, PFAS had the highest concentration in shearwater livers, with an average of around 3 nanograms per gram. The average concentrations of long-chain PFCAs range from approximately 0.1 to 0.6 nanograms per gram. Note that for most compounds, our limit of detection was around 0.05 nanograms per gram. We did not find any differences in the concentrations of PFAS between the two species, except for PFTRDA, which was only slightly higher in flesh-footed shearwaters compared to wedge-tailed shearwaters, around 0.2 nanograms per gram difference. An interesting discovery is that concentrations of PFCAs with odd numbers of carbon atoms are always higher than their next even-chained congener. We asked the question, does plastic ingestion contribute to the overall exposure of PFASs in these birds? To test this, we ran a Spearman's rank test and found that there was no clear correlation between PFAS concentrations and the massive plastic ingested by each bird. This is evidence that the contribution of PFAS exposure from ingested plastic is likely to be negligible. The main source of PFAS to these fledglings is thought to be through the food that is fed to them by the adult. And when you compare the concentrations found in these juvenile birds with others from around the world, we concluded that the area surrounding Lord Howe Island, where the adult birds are foraging, is typical of a background concentration that are only impacted by the long-range transport of PFASs through the atmosphere and through the oceans. This study will serve as a reference for PFAS contamination in birds from Australia, and we hope that regulators in this country will use this information to help prepare future guidelines for the compounds that were detected, but also for the compounds that were not detected. We also asked if the concentrations of PFAS were potentially impacting the body morphometry of these birds. Again, we looked at the Spearman's rank test and found that there was correlations between the concentrations of PFDA and PFTRDA with the size of the birds. When this is plotted, there is a clear positive trend in the data. As concentrations of PFDA and PFTRDA increase, the body mass and wing chord length also increase. There are 2 possible explanations for this. First of all, the birds that were fed more were able to grow larger, and that also increased their exposure to PFASs, which makes perfect sense. But there's also another possibility that these compounds are actually impacting the physiology of these birds. PFTRDA, for example, has been shown to disrupt the function of cortisol in birds from the Arctic. Decreased levels of cortisol can have the effect of increasing body size, which would also explain the results that we see in our study. At the end of the day, correlation is certainly not causation, and more studies into these observed effects need to be done. There may also be other contaminants impacting body morphometry that were not monitored in this study. But there are definitely lots of researchers out there looking at sublethal impacts of PFASs, and I suspect we'll have more answers to these questions in the future. In the meantime, we will continue to monitor this population of shearwaters for potential impacts of emerging contaminants such as PFAS.
And this is now a fairly well-studied species. Then something new and completely unexpected happens where I'm just kind of left literally wandering around in the dark going, where have my birds gone and what has caused this? And sometimes I legitimately don't know.
At this point, I would like to acknowledge my collaborators in this project. My supervisors are Brad Clark, Mark Green, and Raoul Mulder from the University of Melbourne, and Jeff Shemetta from RMIT University. And last but not least, Jennifer Lavers and her amazing team at the University of Tasmania. The Adrift Lab were responsible for all of the field work on Lord Howe Island, in particular Ian Hutton and Alexander Bond, who sampled in 2019. Please check the links in the description for more information. Melbourne Water and Water Research Australia helped fund my PhD candidature, and the Detached Cultural Organisation and L. Bryce generously support the fieldwork on Lord Howe Island. Fluorosulfonic acids, perfluorocarboxylic acids, fluorotelomere carboxylic acids, perfluorophosphinic acids, perfluoroalkane sulfonyl fluorides.